Hi, thank you all for being here today. We are partnered with SRAP, the Socially Responsible Agriculture Project. Here's our lovely team. And before we get started, we'd like to define a few important terms that will help set the stage for what we're going to be discussing today. Within the animal agriculture industry, large-scale facilities that raise high numbers of livestock, in our case cattle, are called concentrated animal feeding operations, or CAFOs. Because these sites house so many animals in such close quarters, they produce a lot of manure, which is just as glamorous as it sounds. To manage this waste, many of these CAFOs use systems called digesters, which break down the manure and convert it into a usable methane fuel. CAFOs are also known to frequently use antibiotics on their livestock to prevent the spread of disease as well as treating illnesses. Over time, this heavy use of antibiotics promotes the development of antibiotic-resistant bacteria, or ARBs. Here's how this happens. So when bacteria is exposed to antibiotics, most are killed, but some mutated resistant bacteria remains. And these resistant strains can then multiply and transfer their resistance to other bacteria. Eventually, they can make their way into the surrounding environment via air, water, and soil. This brings us to our research. We investigated the presence of antibiotic-resistant E. coli in surface water near CAFOs in Tulare County, California, which is home to 11% of the country's dairy digesters. This issue goes beyond environmental health, but it's also a concern of environmental justice. Communities that surround these CAFOs are predominantly lower income, Hispanic and Latino, and made up of farm worker populations, which are groups often facing greater health and environmental concerns and burdens. We conducted this research under the guidance of Dr. Jennifer J. and PhD student Yuhei Zhang, whose guidance and support helped bring our project together.
So for our research project, we had two main topics. The first was to compare resistance profiles in E. coli in surface waters and compare them in different proximity to CAFOs and digesters. And the second was to assess IDEX Coiler 8 as a potential screening tool for multidrug-resistant E. coli, or MDR-EC. And just to note, multidrug-resistant E. coli means that E. coli is resistant to 3 or more antibiotics.
Okay, so on the screen now, you have two different maps. The one on the left is just showing you where Tulare County is relative to the rest of California. The one on the right is showing you where our 10 sample sites are in the blue stars. Those red diamonds are going to be where your digesters within Tulare County are, and then the gray dots are the CAFOs. Tulare County is going to be the right side of that border. So we chose these sites, and the choosing of these sites was super important and also a little bit difficult. They had to be near a waterway, and that waterway needed to have water, which definitely limited us sometimes. Additionally, it needed to be accessible from public land, so not somebody's farm or agriculture facility. So something like a public park or a bridge or a highway. And so, although there aren't a lot of sites, each site had a multitude of isolates that we took from them in order to study. So instead of having a more regional overview, we were able to take a deep dive into each one of the sites.
Like Caitlin just said, we sampled from 10 different waterways near CAFOs. On our sampling trips, we took pH, temperature, and turbidity. Each of our sample trips we took had different numbers of sites we took from because of seasonal variations and conditions out of our control. So after we came back to the lab, we had to do some further analysis. A lot of these tests were time sensitive, which meant that this all happened in one day. The sampling team would leave at 4 a.m. and then the lab team would be in lab until 9 p.m. So we had some really, really long days. We also tested for nutrient and connectivity and prepped for IDEX and our MDR tests. So the first thing I'm going to talk about is IDEX analysis, which is a one-day test. So we tested 5 antibiotics at both intermediate, which was 10 microliters, and resistant levels, which is 100 microliters. We tested these in raw solution, 1 to 10 dilution, and 1 to 100 dilution. We poured each of these samples into the trays that are seen on the left, and then incubated them for 24 hours. After this, we calculated the most probable number of total coliform E. coli and calculated the percentage of antibiotic resistance for individual antibiotics. We also did traditional MDR testing, which was a lot longer, a few months compared to a few days. The bulk of our project was done in the lab, but we'll spare you the hours and hours we spent every week in our lab. So for each of the sampling sites, we isolated 80 colonies, and for each of those colonies, we had to purify 3 times, which is shown in the picture above. After that, we had to test for viability of E. coli by doing an indel test. And then after that, we performed a Kirby-Bauer diffusion test, which is shown on the bottom. We tested 11 antibiotics and measured resistance for each isolate for every site.
So these are our results. On the bottom there are all our samples grouped by the location of them, abbreviation, and then colored by the sampling date. So if they have the same color, they were collected on the same day. The higher the bar, the higher percentage of the bacteria collected in the water has multi-drug resistance. So you can see on the left is our clean site. These were collected in the Sierra Nevada, far away from the farming communities, and they show our expected results for bacteria, 0% multi-drug resistance. Meanwhile, on the right, you can see lots of different levels of multi-drug resistance. Some are up to almost 30%, others are just a few percent, but these are just a subset of our samples. We have more farming samples that are not hidden on this graph, and all of them show some level of multi-drug resistance. So yeah, we have a problem in our farming communities. Now that we found multi-drug resistance, we also realized that finding MDR was a really labor-intensive task, as Emmy talked about. So the second part of our research project was, what if we use one antibiotic to predict multi-drug resistance? So this leads to our second graph. Each line represents an individual antibiotic, and we want to see if the resistance of an individual antibiotic can predict multi-drug resistance. And we only had one antibiotic that had a strong relationship, that is TE or tetracycline. The light blue line over there had an R-squared value of 0.8 and a p-value of less than 0.01. So a statistically significant correlation. And this graph is using the diffusion method. We did the same thing with the IDEX method. The x-axis is the percent antibiotic resistance using IDEX, and the y-axis is multi-drug resistance. And again, TE, tetracycline, the light blue line, had the strongest correlation. The other antibiotics did not show this strong correlation though.
Okay, so our final conclusions, MDR E. coli was absent at the clean sites, but reached up to 29%, as Lawrence just said, at our CAFO impacted sites. And end goal, we would have liked to be able to have a modified IDEX protocol that would be a quick way to test for multi-drug resistance. However, we weren't quite able to do that due to our small data set. With more data, modified IDEX may become a cost-effective and scalable tool in the fight against antibiotic resistance. Again, there are more antibiotics to be tested and more data to get, so we're super excited to see what could happen in the future. And lastly, TE resistance strongly predicts MDR E. coli. So again, more testing is needed. We do think that there is still an opportunity for IDEX to become a standardized field practice to quickly test for multi-drug resistance in surface waters. And this is just going to help streamline water quality testing in these CAFO impacted areas, and also just bring light to the public health problem that is occurring in these areas. We'd like to really thank Dr. J and Yuhui for their support and mentorship in the last year. We're incredibly grateful for this experience.
Excellent. Any questions from the audience? Yeah, please.
I just have a quick question as to what was the consideration of the antibiotics that were chosen? Are they just the most common, or is it just is there a specific reason why you chose the ones that you tested?
So we performed our first round of NDR results, and the ones that had we plotted each of the individual resistance against total MDR, and the ones that had the highest resistance are the ones that we chose to test in our next round for IDEX.
That as well. One of the antibiotics that we used was CTX, or we also it's an antibiotic that specifically the World Health Organization recommends to test for beta-lactam protein-like production in proteins, which is one of the most concerning kind of public health concerns that we're seeing in CAFOs and in other watersheds, not only in Tulare County but also around the world as well. And so we mainly were using that as a control to try to see if that correlates. And as our data showed as well, CTX, which was recommended by the World Health Organization, is not that good of a predictor for other antibiotics as well. So we mainly use CTX as our control, but we were testing a variable amount of antibiotics, and it's still increasing. Dr. J and you are endlessly trying new antibiotics that are up and coming in the CAFOs industry.
The question in the back, yeah.
So, are you planning on publishing this? Is this gonna be submitted as like a research paper?
Yeah, some of us hope to continue the project over the summer to just keep working on the paper and publish it, but that's still variable depending on if the company or get more money. But some of us will volunteer too, so we'll see how it goes. We'll talk with you and Jay about it.
There's another question right here, yeah.
How fast was projects then published out?
Sorry, the question, how fast was the IDEX converted? Yeah.
So IDEX are just one day. We put them in the incubator for 24 hours, and then we could calculate that number right away. MDR, we had to go through many rounds of purification and then like preservation and a bunch of other protocols to just get those same results or like similar results.
Like how many days?
For MDR, do you know?
I mean, I think that we could get it down to 7 active days. However, with availability of students and like midterms in life, it definitely took longer than that at times. Also depended on the amount of sites that had water after a sampling trip. So if we were planning for 6 sites, but only 4 of them, or if we were planning for 6 and then we ended up with 8, that would affect the amount of work that all needed to happen at the same time. And each one, again, like we were doing the process of many steps that Emmy just described for 80 different isolates and then multiply that by the amount of sites that we collected for that trip. So active work, you could probably get it done in a week. We definitely didn't get it done in a week every time. By the end, we were definitely more efficient though.
And then to add as well, I think one of the most concerning things too was that when trying to estimate how much of antibiotic resistance was actually being reported in Tulare County, there wasn't much that we were finding. And so one of our goals of our project as well is that in hopes in validating the IDEX method, we would be able to recommend to scientists and other public health advocates that there is an innovative method out there that can be used to identify the problem, to quickly address it, rather than spending so much time in lab and expecting communities within Tulare County to try to investigate this when we can provide this method for them to actually tackle the problem itself.
So, thank you so much, team. Nice job.